Okay, so UM Wi-Fi is not playing nice with me. I cannot log in with my iPad, so I cannot draw. Um, so I hope you guys can draw on your own. Bit difficult, but yeah. Okay, let me try one more time. If I can't make it, then I will just continue. I cannot log in. Try this. Okay, as you can see, can you guys see anything? As you can see, can you guys see anything? As you can see.
No.
Oh, it's there, but it's very slow. So I do hope that you guys. Hello, it's okay. I haven't started. UM Wi-Fi is not being nice. It's very, 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 very slow. Um, so what I would do is I will go to my office and give a lecture from my office, but you guys stay here. Because my office is just somewhere at the end, the other end. Um, it's not playing nice. See, it's not changing the slide at all. Let's see. 6, 7, 8, 9, 10. Never mind. Okay. How do I hold on one sec? I have one alternative, but hope it's all right. Come on. Am I? Okay, I have to do it manually. My drawing will be very, very, so you have to bear with me. Because I need to draw using touchpad. 2004. Let's one. This one. Where is this one? Here. Okay. Let me share my entire screen. Take over. Group. All right. So good afternoon. Unfortunately, I cannot use my iPad. EM Wi-Fi is not playing nice. So what I'll be doing is I'll just be sharing the PowerPoint slide from my laptop. Therefore, I won't be able to draw anything. Wah, see, suddenly ramai orang. Yay. Alright. Can you guys hear me okay? Or there's a delay? Anyone online? Can you guys hear me?
I think you're quite lagging, Doctor.
Yeah, it's lagging, Doctor.
It's UM Wi-Fi. I blame you, Wi-Fi. Hopefully, otherwise you should have come here. Okay. In this subject, a lot of problems. Play, guys. I think I have to move to my office. You can see it's very, very slow because UM Wi-Fi in this room is very, very weak. Sometimes it's okay, most of the time it's not. Boleh tak? You guys want me to be here? Or right. So what I will do is I'll give you guys so that you can see your friends, and I will rush to my office and I'll do my lecture from there. Can you guys hear me clearly? Thumbs up if you guys can hear me. Can you guys hear me?
Yes.
Yes.
Okay. But those who are at the auditorium, can you hear me? No? Tak clear? I don't want you guys to go and online. See, my internet at my office is better than the one at the auditorium. For whatever reason, what would be the best way to do? Or is it still laggy? Alright, never mind. We'll just have to go through because otherwise we are wasting more time again like last week. Okay, or perhaps someone can bring a mic. Yeah, thank you. Or you can increase my speaker volume. Do you know how to increase speaker volume on the Mac? If you look at the keypad, there's a volume icon. So you can just drag the volume icon if you do see that. So can you hear me now better? The people at the Audi or not? Okay tak? Okay tak? Testing, testing. Can you hear me okay? Okay. So one second again, because I need to make sure that it's recording. Which actually is this? Okay, those at Audi, can you hear me? I cannot see your face anymore, so someone need to come to the front and tell me whether you can. Boleh? Okay, alright. Just do this quickly so that I can make sure that I will record the lecture. Okay. My PC. All right. Now I can see you guys. At the same time, I can see me. Okay, it says they're recording. All right. So last week we stopped a bit early, and I think we stopped somewhere here. Okay. So we were talking about how we are talking about how we can actually, or how amino acids are made, and I've talked about there's 20 amino acids, naturally occurring amino acids. There's some literature mentioned that there's about 22. It's debatable, but nonetheless, everybody agrees there's a minimum of 20. Okay, so 20 is just considering the L isomer. We're not talking about D isomer at all. If you do consider D isomer, then it will be more than 20. Okay. As I mentioned, it's no longer a fact, okay, because we do have bacteria that produces the amino acids and consumes and utilizes the amino acid, and an antibiotic from fungus that utilizes again the amino acids. So this is again a rough number. It's not a kind of like a fact anymore. So a long time ago, people always believed that there's only 20, but now it's no longer 20, even though the numbers are still not changed. Okay. So a combination of amino acid resulted in a protein of different characteristics and function, and of course, depending on the length, you can have a short amino acid, a short protein, or a long protein, and each an individual protein can have a unique characteristics. Okay, so in general, in humans, we have 10 essential and 10 non-essential amino acids. So why do we call it as essential? So this is because our body cannot produce it in enough quantity, or some of the amino acid we can't even produce it. Therefore, we require this amino acid. It's very essential. It's very important for us to require to acquire this amino acids. And the only way by which we can acquire the essential amino acids is by obtaining from dietary. Okay, so we can only eat, and then by eating we can get all these amino acids. Of course, you can always inject your amino acids into your body, but nonetheless, eating is easier and it's more fulfilling, right? And how about non-essential? Non-essential are the amino acids that we can produce in our body. It is still important for growth. Doesn't mean that when we say that it is non-essential, it's not important. No, the essential here refers, it goes back to our, I would say, grandfather generation, by which they say that the word essential and non-essential meaning to mean that this amino acid can either be produced by our body or cannot. So the essential ones are the ones that we cannot produce. Thus, it's very important for us to take from an external sources. So essential, while non-essential, our body can produce it in our own cells. Okay. Source can be from essential amino acids or even our food. So as you will see in the next few slides, we'll show you how our body actually produces amino acids. So these are the amino acids. So you can see non-essential, there's about 10. Non-essential is also about 10. Okay, so if you want, you can try and identify what is the last non-essential amino acids. It's not critical, it's just something that if you want to play around with. Okay, so why is R here in bold? Because R is an amino acid that our body can produce, but our body cannot produce it in sufficient quantity at a certain stage. For example, for the ladies, at one point in time, you might get pregnant. So during pregnancy, your body cannot produce enough arginine or R amino acid, so that it will cover both what you need and what your baby need. Okay, so that's why R is still listed in an essential, even though our body can actually produce it. So that's a few arguments on why we need to label this essential and not essential. It's not really important, but what I want to introduce here is that so that you know, some amino acids are consumed largely from our food intake, while others we can actually produce it. So if you are thinking of buying vitamins, for example, as a supplement, okay, so what you might want to do is you might want to look at the vitamins with amino acids. You might want to look at the sequences and see whether the vitamins actually is indeed needed or it's not. Okay, one example is if you look at vitamins for kids, especially for primary school children, you can see that nowadays vitamins has lysine amino acid. Okay, so this one is lysine. Okay, lysine amino acids added as part of a vitamin. So, and you can see here that lysine is part of an essential amino acids, meaning that our body cannot produce. Therefore, if the younger kids actually consumes this vitamin with lysine, the lysine will be absorbed and thus be utilized in your body and in their body. But say the vitamin says it has A and D added amino acids, then you know that it's just a selling point. Okay, so by knowing this, you can actually think about whether the vitamins with added amino acids are really what you need, or you don't really need it at all. Okay, so a good judgment. All right, so this is basically how organisms, not just us, organisms produces amino acids. If you look at here, there are, I think if I'm mistaken, I've calculated about 18 amino acids in total from this page and the following slide. Okay, so these are just a step just to introduce you guys on the just to tell you guys that we can actually produce amino acids. I'm not just giving you guys lies. Okay, but at the same time, you do not have to memorize this. Okay, I never actually asked in any of my quiz or lectures what is how many steps, for example, how many steps for you that you need to take from 6G6P to produce 3PG. Okay, I never actually asked that. So this slide is just to showcase what are the processes, what are the steps by which you can produce amino acids. Okay, and the one down here, okay, if you see it's pyruvate, this is an alpha keto acid. So you can see here, this is an alpha position from the acid. So we call it alpha keto because it's a ketone, alpha keto acid. And you will see why this step is very important, especially in a chemical synthesis of amino acid. Okay, so this is just a continuation. So there are three different steps, three different processes, main processes. Obviously, steps-wise are more than three. There are three main processes. If you see here, one is what we call as citric acid cycle. One is a glycolysis, and the other one is phosphate pathways. Okay, just for you guys to know. All right, and that is all. These are just a bit of summary, references, and that is all for today. I'm joking. We have another lecture. We're gonna continue with lecture number 2. All right, so you can see here, first slide is about whole F. So please open your devices. Those who are actually at the auditorium, perhaps if you're lucky, you can get UM Wi-Fi. If you're not lucky, then hopefully you can use your mobile. pole F is not that heavy, but then nonetheless, the internet in over here is a bit weak. Okay, all right. Let's go and see the first question. Before that, I need to open my poll everywhere on my desktop over here. Give me 1 second. Okay. 405. What is the central dogma in molecular biology? You should be able to see the question now. Okay. So what is the central dogma of molecular biology? So this one is based on last Thursday lecture. It's the same lecture notes as what we've seen previously. But yeah, it's just a basic question. So what you need to recall what does it mean by central dogma. So what does it mean? Central dogma of molecular biology, of course. So definition of a central dogma is something that it's kind of like people believe is the truth. Okay. So central dogma. So when we say central dogma of molecular biology, then it refers to what do people believe is true about molecular biology. So the first one is DNA to RNA to protein. Second one is RNA to DNA to protein. The third one is protein RNA DNA. And the fourth one, protein DNA RNA. Or third one is RNA DNA. And the last one is none of the above. Okay, what is the central dogma of molecular biology? Okay, if I don't see any changes, I expect that this is the final answer. Okay, so to say the least, number one is the correct one. Okay, so even though it's no longer a fact, but at one point in time, that is the central dogma. So you have DNA, DNA produces RNA, RNA produces protein. Okay, so DNA to RNA, this is what a process that we call as transcription. Transcription, while RNA to protein is what we call as translation. Okay, so one is transcript, one is translate. So this is the general central dogma. Of course, if a question like this comes out in your final exam, I will also accept none of the above, because the central dogma is no longer a kind of like a real fact or a real belief, because now we know a lot more. Okay, number two, there are a total of recognized amino acid by the IUPAC. So there are a total of how many amino acids being recognized by the IUPAC system? Okay, choose the closest one that you think is correct. Okay, and give you guys about 5 more seconds. Oh, it's still changing. Okay, done. Final. Okay, in total, I just knew that funny thing, because I'm actually opening IUPAC system, and it recognizes 23 amino acids. So there's no answers. Okay, the closest one is 22. Again, 20 is not the number of recognized by IUPAC. 20 is the number of amino acid universal in all organism. Okay, so 20 is universal in all organism. All organism has a minimum of 20 amino acid, but the IUPAC, if you actually just Google IUPAC amino acids and count apparently there's 23. So there's more than 22 now. So there's 23 something. Oh wait, it is 22. The other one is called XAA, which consists of all amino acids. Okay, so 22 is a specific, and then the third one is none. So basically the correct answer is larger than 22. So minimum of 22. Okay. All right, I need to remember this so that I can update the lecture notes for the next semester. Number 3, non-essential amino acids can be obtained from consumption. True or false? Okay, non-essential amino acids can only be obtained from consumptions. There's only two answers, true or false. There's no I don't know here. So you need to choose. Even though our class today is way less than this morning, but it's still okay. It's always hiccups in the afternoon lecture. Either some of you guys are sleeping, doing something else, or in our case, because some of you guys are actually in auditorium, no proper internet connection. Okay, all right. So I think I give you guys enough time. The correct answer is false. Okay, non-essential amino acid can also be synthesized in our body. Okay, you need to remember essential is the one that cannot be synthesized, so you need to eat. Non-essential is the one that can be synthesized and you can get it from eating. Okay, right? So, and the last question, oh, this is the last question. Okay, all right. So, I think I have another question. Just keep your mobiles handy. Do have another question. I think in the middle of the lecture notes. So that is the LO. Amino acids continue. Just to cover a little bit on the importance of amino acids. So in the industry, in Malaysia and elsewhere, these are the main four how to say industries that consumes, that utilizes amino acids. So we have food, supplements, poultry, and cosmetics. So food, the most common thing that probably you guys heard multiple times is MSG. Okay, what is MSG? MSG is monosodium glutamate, and based on, if you have remembered all 20 amino acids, glutamic acid is one of the amino acids. Okay, so you can actually produce MSG very, very easily. I can produce it in my lab because I do have glutamic acid. So what you do is you have glutamic acid, you put in sodium chloride, you mix them together, put some water, then you can produce MSG. Okay, very, very simply and very, very easy. And amino acids can also produce a special taste. For example, there is a research paper, so this is not made up notes. Okay, so there's a research paper saying that you can actually produce an artificial flavor of a snow crab. I have no idea what snow crab is. I never actually eaten a snow crab, but if I were to actually taste how snow crab tastes like, I need to mix alanine, arginine, glutamic acid, and glycine together. I'm not sure about the ratio. I need to reread the research paper, but they mentioned that this four amino acid produces a flavor similar to how if you are eating snow crab. Okay, I have no idea where you can get snow crab from. Supplements, we've looked at drugs this morning, where you can actually design drugs using peptides. So peptides can be produced from amino acids. Okay, antibiotics, for example, gramecidin D is one of the antibiotic that is made from amino acids. Yes, it's not just the 20 amino acids, it's in combination with other amino acids, but nonetheless are amino acids. And of course, vaccines, for example, HPV vaccine, HPV, or our COVID vaccine, not the Pfizer or AstraZeneca, but the ones that utilizes the COVID-19 virus itself, like Sinovac, for example. So you need to actually feed in amino acids with your um, with your cells, so that your cells will consume the amino acids, and the amino acid will then be used to produce more COVID-19 virus, and you can kill the COVID-19 virus and utilize that as a vaccine. Okay, poultry is mostly supplements for animal feed, because if you have a big poultry, you might not have enough grass or fruits, for example, to provide to your animals. Therefore, you give them in the form of supplements, normally injection, or you can actually mix and produce foods um, for your animals. Okay, and last one, amino acids is used as an additive in cosmetics. For example, for hair care products, normally they utilize the cysteine, because cysteine has a sulfur. Sulfur can be a substitute for what's the sulfur-based reagent? I can't remember on top of my head now. Okay, so normally if you look at your shampoo, there's this one ingredient that contains sulfur, and it is actually bad for your hair. So cysteine can be used as a substitute. It's less harmful, but it does the job in the same manner. Okay, and also in cleansing agent, for example, glutamic acid and glutamine is being used in what do you call it, face mask, not our COVID face mask, the where ladies normally wear them before they went to bed. Okay, so that type of face mask. All right, so we've looked at just now on how you can actually produce amino acid via in the cells. Okay, we've looked at the citric acid cycle, for example, that utilizes a lot of enzymes to produce amino acids. And from there, again, I just want to highlight about the alpha keto acid. Okay, just remember that particular structure, because that is one of the reagents that we need to actually produce amino acids chemically. Okay, so how do you produce it? There's multiple ways. So one of the basic way is to do an acid and base hydrolysis of animal proteins. Okay, because based on last week's lecture, you do know that proteins is made up of amino acids. Therefore, if you hydrolyze proteins, you can get amino acids back. Okay, so that's the easiest way to do it, and that's how our body actually gets our amino acids. Okay, we eat all meats, chicken, beef, or eggs or beans, for example, that contains a lot of proteins. We eat it, and then in our stomach, there's a lot high concentration of acid. You hydrolyze all the proteins. We do have enzymes in our stomach as well that helps in digesting these proteins, and therefore it goes to your intestine and it will be absorbed into your body. Okay, sorry, I want to drink. And also decades ago, just doing a simple hydrolysis is not the best way, because imagine if you have a protein. Okay, even say the protein is tau protein from fruit fly. Okay, so tau protein contains 14 amino acids. However, the 14 amino acid is a mix of amino acid. It's not just alanine throughout. It's a mix of alanine, glycine, blah, blah, blah. Okay, so if you were to just use hydrolysis as the only mean to produce, to acquire amino acids, that will be a bit difficult. Why? Because towards the end of the day, if you just simply do hydrolysis of proteins, you need to add in purification. Okay, because if you don't purify it, then you cannot get the exact pure pure amino acids. Okay, so scientists decades ago found out that there is a synthetic pathway that you can actually utilize and get a pure amino acids. Okay, and in some cases that we will see in the next few slides, there are an unusual and unnatural enantiomeric amino acid is needed. Okay, so we will just look at it as in some examples. And most critical for a synthetic asymmetric amino acid synthesis is to acquire an optically active form. Okay, again, because in our body, the 20 universal amino acids are from L amino acid, not D, but the L ones, the L isomer. Therefore, we need to produce, find a way to actually produce, and thus you can actually make it. You can make amino acids in the lab. Okay, so there are multiple approaches, but these four are the chemistries that are widely used worldwide, even nowadays. Okay, why don't people switch to a different mean? Because this, as you can see later on, is very, very simple and straightforward. Okay, so I have no idea if people can manage to produce a better way other than using biology. So of course, if you are doing via the biology pathway, then you can get a lot of a higher quantity perhaps. Okay, but nonetheless, for now, chemical pathway are still used. Okay, so four widely used, and we will look at number one, which is Helvohard Zielinski. So I will just abbreviate that as HVZ. Okay, so HVZ can be utilized as a first step to produce halogenated alpha carbon of a carboxylic acid. So that's what you want. Following reaction with ammonia will produce an amino acid via a substitution reaction. So recall that halogen is a good leaving group. So a few things that you need to recall is halogen is a good leaving group and a substitution reaction. Okay, is it SN1 or is it SN2? Okay, recall back what you've learned previously. Of course, it's not E1 and E2, but just by learning this, I hope that you can at least recall a little bit what does it mean by SN1, SN2, E1 and E2, and so on and so forth. Okay, so this is how HVZ reaction works. Okay, you have a carboxylic acid. You react it with tribromophosphine in the presence of a bromine, and then you hydrolyze it with water. You will produce an alpha substituted bromo acid. Okay, and then in the presence of excess amine, you can actually remove the bromine and thus get an amino group to your structure. So this is the easiest way to produce amino acid. Okay, so what are the chemistries behind it? It involves four key reactions. Number one, nucleophilic acyl substitution. Number two, keto-enol tautomerism. Number three, bromination. And number four, hydrolysis. So let's look at these four reactions in a bit more detail. Okay, in case if you need to refresh a little bit, you can do it at home. Thinking about what does enolation means, what does tautomerism means, a little bit of pKa and acidic proton. Okay, so these are the keywords specific if you want to go deeper in this reaction. All right, so how does it work? So the first reaction, if you look at the previous slide, shows that first one is you need to react it with tribromophosphine with bromine. Okay, so what does it do? This reaction pretty much first off is to create that one. So of course you have two alphas hydrogen. Okay, so the first one is to create a bromo acid. Okay, acid bromide, bromo acid. Okay, to create an acid bromide. See, so this is the first reaction. But in excess, what you will get is you will also get a substitution of one of the alpha hydrogen to another bromine. Oops, sorry. So you will get that. It's one line with alpha hydrogen, the other one with bromine, and then you can still get the bromo acid. Okay, and then this is where step number two comes in. where you will hydrolyze your bromo acid, and thus you'll get a alpha bromo substituted acid. Okay, and notice over here is a racemate, because again, if you looked at this particular structure over here, so the bromo substitution can come from either direction, therefore you can get a racemate. Okay, and from the racemate alpha bromo acid, in the presence of an excess ammonia, so ammonia is a nucleophile, so it leaves nucleus and thus attacks at that particular alpha carbon position, and bromine will go out as a leaving group. Okay, and because you started off with a racemate, and thus your product will also be a racemate. Okay, so the resulting alpha bromo carboxylic acid can undergo a SN2 reaction and form a racemate DNL amino acid in the presence of excess ammonia. Okay, so again, I'm just saying bromine is a good living group. That's one thing. And then you need to recall SN2 reaction. Why do we call this as SN2 reaction? It's because you need two entity, you need both entity to be present at the same time. Oops. Okay, and this one is the acid. We'll just put it acid. Okay, in order for you to get your product. Okay, so that's why we call it SN2 reaction. SN1 reaction, on the other hand, is the formation of normally carbocation, which in this case it doesn't. Okay. All right, so I'll leave you guys for you to fill this up if you have not, but if you have, then it's very straightforward. It's kind of like a practice. If you want me to check it, then I will check it. I will give you guys the answer next week if you need to, or perhaps I can just repost this later on. Okay, so that is all about HVZ. Moving on to the second reaction, which is reductive amination. Perhaps we can cover this within 5 minutes, and then I will stop there. Okay, so the reaction is called as reductive amination, or you can it is also called as reductive alkylation. So why reductive? Because it involves a reduction process. Okay, particularly at this. See, it's a one-step catalytic reaction to form an amino acid when an alpha keto acid is treated with ammonia, then hydrogenated in the presence of palladium catalyst. An imine intermediary is formed prior to the catalytic hydrogenation. Okay, so again, this is the alpha keto acid. If you recall, when I mentioned that, please remember, in the pyruvate slide, there is also an alpha keto acid. Okay, so it has the same structure. Okay, so this is why biology and chemistry are more or less the same in terms of reductive amination. Okay, so there's two steps reaction, even though it's one part, but it's a like two steps. First one is you react it with an excess of ammonia, and then you turn on a palladium catalyst in the presence of hydrogen. You bubble it up, then you'll get your amino acid. Okay, how does what is the mechanism? So the mechanism is, again, you have an ammonia. Ammonia attacks at the alpha carbon position of the ketone. The ketone picks up hydrogen, and thus some electronic rearrangement. The oxygen will leave as water. Okay, and therefore you will get an imine. Okay, so alpha imine. So that's the first step of the mechanism of the reaction for reductive amination. And then followed by that, once you have a palladium catalyst, you bubble it with hydrogen. Hydrogen promotes hydrogenation, breaks down double bond to form hydrogens to form a single bond with hydrogens. Similarly, similar concepts applies here. You have a double bond, an imine double bond at the alpha keto position. In the presence of catalytic reaction, then you'll get your amino acids. Okay. Basic one. All right, so another example, if you do change the alpha keto acid, so in the previous slide, it's just a general alpha keto acid. Over here, you do have a 3-methyl 2-oxopentanoic acid. You can then react it with excess of ammonia, forming 3-amino. See, you can see there, amino 3-methylpentanoic acid. And then in the catalytic hydrolysis, not hydrolysis, sorry, catalytic hydrogenation, then you will form your amino acids. So in this case, this is I think isoleucine, if I'm not mistaken. Yep. Okay. So then you can produce amino acid. So as you change your starting material using this same process, you can form the amino acid that you want. Okay. But again, okay. So I'll just stop here. This is the last slide. Go back and open your poll F again, and just let me know. In this process, reductive amination, the product is it an L amino acid, a D amino acid, R amino acid, or S amino acid, or racemate? Okay, based on this reaction, what would you get here? Is it an L, D, R, S, or racemate? I have a few answers here. I have 9 responses, I think, for now. Or 19, sorry. 19 responses. Okay, so LD or racemate. If I count to 5 and there's no changes, then I will stop. 1, 2, 3, 4, and 5. Okay. All right. So the answer is racemic. Okay. So why racemic? Because again, if you look at here, this is a flat surface. Okay. So it's a planar, not flat. It's a planar. So in the presence of your palladium catalyst, so palladium catalyst, if you want to use it, it's normally you use it as a small chunk of palladium. So the surface contact between the intermediate with the palladium can come from either this side or that side. Okay, so depending on which side is the contact side, then you can get either L or D. Okay, so the answer is me. Okay, all right, so that is all for today. We'll stop here. I'll try and figure out what we can do next week. Worst case scenario, we will move back to DK Vanda, since it's a bit hard for me to actually give a lecture even in our auditorium. Okay, that's all from me. If you do have any questions, you can ask now, put in the chat, or you can ask in the WhatsApp. Okay, that is all. Just one last thing for your continuous assessment. So I mentioned that you need to work in pairs, right? 2 in a group. So I do not have any Excel sheet by which you can register and register who are your group members, because there's only 2%. So I think the collaborative work between you two will be much higher than working in the 4 or 3 in a group. Okay. And of course, because you just need to choose which topic, you don't need to tell me. What I need is once you submit it, that will be your topic. Okay, that's all from me. Thank you and have a good day. You guys might want to go back early, since it looks like it's a bit cloudy and it's going to rain soon. All right, take care, guys.
Thank you, Doctor.
Thank you, Doctor.
Thank you.
Welcome.
Thank you, Doctor.